All right, so our last unit is chapter 28 and chapter 29. And so you'll see in chapter 28, we'll be talking about the reproductive system, the structures of the reproductive system. We will begin actually with a little bit of kind of almost like an intro to genetics, but genetics will be part of chapter 29. And then chapter 29 will be covering what happens when the sperm and egg fuse. So it's going to be focusing on human development, then a course of pregnancy, and then the very last unit will be on genetics and heredity. And so you will have some assignments here. It's almost like we started with a lot of assignments, right? The first unit and first two really units were kind of heavy on assignments. You had a lot of those lab things, lab exercises for practice. This last unit is going to be kind of similar. So we will have some things that you will be turning in as far as the lab exercises. And this is because you will need to practice some of those genetics problems. So just make sure that you're paying attention to those due dates, okay? And then of course you will have some visible body quizzes there. So make sure that you're also looking at the modules in the visible body. So don't just go into the quiz. The modules should be done first so that you get ready for those quizzes. So let's go ahead and start talking about the reproductive system. And I will split this into, I think, three parts. So today we'll do the intro and meiosis. Then you have the recording that's currently on your MyTCC. There's only one right now. That's one that I've done a few weeks back, actually. But that's it's. I would just say the same thing, right? So that one is on the male reproductive system, and then I have a section on female reproductive system that I will be uploading for you as well. So let's go ahead and begin with chapter 28. So reproductive system is a system that is responsible for making sure that the species continues, right? It's responsible for reproduction, but it has some other functions as well. We'll see that some of the hormones that are associated with reproductive system are going to be responsible for development of secondary um, characteristics, right? And so anyway, but the main organs of the reproductive system are called gonads. Gonads is a general word for reproductive organs. And in males, gonads are testes. In females, gonads are ovaries. Okay, so these collectively are called gonads. The reproductive organs, or these gonads, are going to produce gametes, and they're also going to produce sex hormones. If you are not familiar with the word gametes, gametes are your sex cells. And in males, these gametes are sperm. In females, gametes are egg cells or ova. So the gametes and sex hormones is what the gonads are going to do. There are going to be several accessory structures that we will cover as we are talking about reproductive system as well. And these are going to be different in males and females, since these systems are different in males and females. The gonads become fully functional in both males and females beginning in puberty. Although we will see that the gonads of females, which are the ovaries, begin to do something or even before birth, before the female is born. But full functionality in both males and females begins in puberty. The basic pathway or the cascade of events that happens when the gonads become fully functional is that the hypothalamus is going to release gonadotropin releasing hormone. Gonadotropin releasing hormone is going to act on anterior pituitary, and anterior pituitary will then stimulate luteinizing hormone and follicle stimulating hormone, and these two will act on the gonads and stimulate formation or maturation of the gametes. Okay, so the sperm and eggs. So that's the general cascade. It's the same in males and females. You start with the gonadotropin releasing hormone from hypothalamus. That stimulates luteinizing hormone and follicle stimulating hormone in both males and females. And then these two are going to act on gonads and promote stimulation of and formation of gametes. The males typically produce about 100 million sperm a day. So each day, the male testes are going to produce about 100 million sperm a day. And on the other hand, when we look at females, females typically produce one and release one oocyte each month. So quite a difference there, right? For males, it's 100 million a day. For females, it's one roughly once a month. And I say roughly because it's not exact month, and this can also change, as we will see when we talk about the female reproductive system. So let's see, let's look at our first image here. And actually, before I go into that, or can I go into that? Before I look at the picture, let's talk about the reproductive system homologs. And you're going to see several words that are going to begin with the homo or hetero in these two chapters. So homo means the same, hetero means different. So when you hear the word homolog, homolog means corresponding structure. Okay, so, and we'll also see homologous chromosomes. These are going to be corresponding chromosomes, which means that they're not identical, but they are corresponding. If we're talking about structures, these structures originated from the same primordial tissue during fetal development, so or embryonic and fetal development. So homologous structures or reproductive system homologs, if you're looking at the table, and I don't have the table in front of me right now, let me see if I can pull up the table. Okay, where are you? Is this it? Yes, this is it. So let me try to share this. All right. Okay, so if you look at this table, table 28.1, it summarizes nicely for you what the homologs are. So the first homologous pair here of structures is the ovary and testes. So what we will see actually towards the end of this chapter is that during fetal development, the cells that give rise to ovaries are the same cells that will give rise to testes. It's just going to be determined whether there is a Y chromosome or X chromosome as far as what these cells become, okay? But the origins are going to be the same during this fetal development or embryonic development. So ovaries and testes produce gametes and sex hormones. So their function is the same. Now, if you were to look at specifics of that function, then that's where the differences are, because the gametes are different and the sex hormones are different. But their common, like, overall function is the same. They both produce gametes and they both produce sex hormones. So that's what the homologs mean as well. Okay, they originate from the same cell line or same cells during that embryonic development, and they will have the same or common function. Another pair here is clitoris and glans of penis. And then as far as function goes, there's another pair that has the same function, and that's the labia minora and body of penis. Again, these originated from the same cells during the embryonic development, and they have the same function, but they do look very differently between males and females. And these both are going to contain erectile tissue, and this one is stimulating during sexual arousal and orgasm. Another pair is labia majora and scrotum. These are protective structures, so they cover some reproductive structures and protect them. And then a couple of glands here: greater vestibular gland in females and bulbourethral gland in males. This one secretes mucin for lubrication in one in males and one in females. So that's our homologs or homologous structures. So just remember, homologous means that they're corresponding. They're not identical, but they are corresponding. They have some things in common. For the structures, it would be the origin and the function, but they look different, okay? So let's look at the other image here that I wanted to talk about. So talk a little bit about some anatomy here, just general anatomy, okay? So now I'm sharing this. And that's this image right here. So here we're looking at the perineum or perineal area here. And you can notice, if you remember talking about the skeletal system, okay, you can notice that the pelvis is formed differently between males and females. I mean, not formed, but the angle is different. And there's some other structural differences as you are looking at this. So you can see in females, the angle there is wider. So female pelvis is wider. This is to accommodate the passage of fetus during childbirth. Males don't have a need for that, so their angle there is narrower. The two areas that we can describe are the urogenital triangle and the anal triangle. And just be aware of what is what here. I'm not actually going to ask you about these muscles here, so you don't need to know that, but be aware of those triangles, those areas, those regions, and know that the angle is different. But I think you already know that from AMP1. So that's that about this image. So we're gonna go ahead and start talking about the meiosis, and that is or gametogenesis, and that's going to employ the process of meiosis. So gametogenesis is a process of formation of gametes. So this is going to describe how the eggs and sperm are formed in us. And you will actually see, since males produce millions of these every day and females only one, there's going to be some differences between this process in males and females, but we're going to just kind of describe the general pathway first. So before we get into that, you have to be aware of some basics of genetics. So humans have 23 pairs of chromosomes. And we said 23 pairs, so that would be 46 individual chromosomes. But we have 23 pairs. 22 of these 23 pairs are called autosomes. So they're autosome pairs. And one pair is going to be the sex chromosomes. And this is going to be generally X and X for female, X and Y for male. There are some variations to this that we are not going to discuss here. But there could be some variations of the sex pair. But generally, X and X is female, X and Y is a male, and that's that. The other 22 pairs are called autosomes. So we call these pairs of corresponding chromosomes or matching chromosomes homologous chromosomes. So again, there's that word homologous. Homologous means that they're corresponding, but they're not identical. Okay, so you have each parent gives you one set of chromosomes. Okay, so you have 23 chromosomes from your mom, you have 23 chromosomes from your dad, and that's what gives you the 23 pairs. Okay, so you have one set of chromosomes from mom, one set of chromosomes from dad, and then they'll pair up. So the homologous or matching chromosomes are that's what that means. It's, for example, chromosome 1 from mom and chromosome 1 from dad would be a homologous pair. Okay, they're corresponding but not identical. Cell that has 23 pairs of chromosomes is known as a diploid cell. Okay, so 23 pairs, diploid, di prefix means 2. Okay, so that's your regular cells. These cells are your somatic cells. They're designated by 2n. And N is going to mean number of unpaired chromosomes. So diploid is 2N, okay? And N is number of unpaired chromosomes. That's basically how we designate this in genetics. There are cells that have 23 single chromosomes, and these cells are called haploid. So haploid reminds me almost like a half, even though it doesn't have the F there. But haploid means that it only has one set of chromosomes, and these are your gametes. They're designated by the letter lowercase n. When I said that each parent gives you 23 chromosomes, right? 23 just single chromosomes or one set of 23, this means that the gametes are going to be these haploid cells, all right? So the gametes are formed by the process of meiosis, and meiosis is known as a reduction, division, or lessening. Sometimes they only refer to that for meiosis too. This is necessary. Meiosis is necessary to produce these cells with only 23 single chromosomes, so that during reproduction you have a cell with 23 single and another cell with 23 single chromosomes. When they come together, then you have your first diploid cell. So every body that produces gametes has to kind of go through this reduction. In humans, it is not beneficial to have more chromosomes. And if we didn't have this meiosis process, if we didn't have our reproductive cells have only one set of chromosomes, but kept them at two, then when they would come together, you would have 4 of each chromosomes. And then the next generation would have 8 of each chromosomes, and that would not work. That is not, our cells or our bodies are not designed to accommodate extra chromosomes. Now, plants, that's a different story. Plants actually can have multiple copies of each chromosomes, like 8 and 16, and it doesn't really mess them up like it would mess a human body up. Okay, so that's, having too many chromosomes is almost always non-compatible with life. So, and I say almost always because there are special cases where it is able to result in a live birth. So, okay, so let's look at meiosis. Let me share the picture with you so we can have a visual of how this works. And this is going to be in 2 parts. In your textbook, it's spread over 2 pages. So you can look at that there. All right, and here we go, I think. No. This one. All right. So here's the image here. So this process of meiosis is going to begin with a diploid cell. Okay, it's going to begin with a cell that has 23 23 pairs of homologous chromosomes. But at the end of meiosis, the whole process of meiosis, you're going to end up with 4 cells that are non-identical and they're haploid. So I want you to write down somewhere the difference between meiosis and mitosis, and I think I'll repeat that after I go over meiosis as well. Mitosis, which is a regular cell division, is going to result in 2 genetically identical diploid cells. That's mitosis. Okay, mitosis is your regular cell division, and it gives you 2 identical diploid cells. Okay. Meiosis is going to result in 4 non-identical haploid cells. So all of those words are different except for the last word, cells, right? So meiosis results in 4 non-identical haploid cells. That's the major difference between these 2 processes. Now you will see some similarities here, as like when you were looking at the phases here: prophase, metaphase, anaphase, telophase. That's the phases that we see in mitosis as well. However, here you see that it says meiosis 1, and so prophase is 1, metaphase 1, anaphase 1, and all that, because we're actually, the whole process of meiosis is divided into 2 divisions. And so you have to specify if you're talking about prophase 1 or prophase 2. Okay. Okay, yes. Can I repeat which one? Okay. So meiosis results in 4 non-identical haploid cells. And mitosis, which is the regular cell division, gives you not 4 but 2. They're identical diploid cells. And so after I go over the meiosis process, I'll say that again, okay? So we're starting with the diploid cell. It's going to look kind of similar to what you would see during mitosis, right? You're going to have a cell that is going to get ready for cell division. And what do we do when we're getting ready for cell division? How did I skip this one image? Where did it go? Let me share something else with you because I want to point out something else. I don't know how I missed it. Oh, here it is. I missed it because it's in my file as a table and not as a picture. Okay, so look at this, okay? A little bit more of terminology that I have missed before we go into the meiosis. So when you look at the bottom of this table, you see a single chromosome, single unreplicated chromosomes. This is what you would see in a cell times 46, okay? So that's a single chromosome number, I don't know, we don't know which one this is, okay, but that's a single chromosome. Now each diploid cell is going to have its homologous pair somewhere to it. So in the middle of that table, you can see that's a pair of chromosomes. They're not replicated, they're just in a regular cell that is not getting ready for division, okay? So you would have 23 pairs like this. However, when a cell starts preparing for cell division, whether it's going into mitosis or meiosis, it has to replicate its DNA. And what it will do when it replicates its DNA is it will do something like this. Okay, I don't know. It will create a copy of this one. I am drawing with my touchpad here. And it will also create a copy of the other one. Okay, it's not that horrible, I hope. Okay, so as you can see at the top there, that's a replicated chromosome. So when the cell goes into cell division, it will replicate every single chromosome. And so all the chromosomes will be replicated like this. So now what I drew in, that's a pair of replicated homologous chromosomes, okay? The red and blue are not identical, but the two blue parts, the original and the one that I drew, those are identical. And then the two red parts, the original and the one I drew in, are identical to each other. We call those sister chromatids. Okay, so sister chromatids are identical. They are a result of DNA replication in a cell that is getting ready to divide. Okay, and you have to replicate the DNA so that the new cell has its own copy of genetic material, right? Okay, so that's what it would look like before the cell divides. It's going to have 23 pairs of replicated homologous chromosomes. Okay, so 23 replicated chromosomes there. So let's go back to the meiosis part. Okay, and here we are. So the process of division of meiosis is going to begin with the diploid cell. And this diploid cell has replicated all its DNA, so it's got 23 replicated homologous pairs. Now, on the image, they're just showing you 3 and not 23. If they put 23 in there, it would get very messy. So just in your mind, know that you're only looking at 3, but in reality, there's 23 replicated homologous pairs, okay? What happens during meiosis 1 specifically, and even more specifically in prophase 1, is that the homologous replicated pairs will find each other. Now, this doesn't really happen in mitosis. In mitosis, the homologous pairs really don't care about each other. They don't care that the other one exists because the whole DNA is getting split evenly into two cells. But here in prophase 1, the homologous pairs are going to find each other, come kind of close to each other. And when they do this, we say that they form a tetrad. Tetra, the prefix tetra is 4, okay? And it's 4 because when you look at these legs, if you were to split the tetrad into individual single chromosomes, you would have 4. Okay, so let me see if I can show them to you. Okay, you'd have one here, one red, another red, that's 2, and this one blue, and the other blue. Okay, so that's 4 individual chromosomes there. You could separate this tetrad into 4 individual chromosomes. And if you remember, I said meiosis results in 4 non-identical haploid cells. So each of the chromosomes in that tetrad will end up in a separate cell. Okay, and that's going to happen with every of the 23 chromosomes, or 23 pairs. Okay, so clear that. And yeah. So in prophase 1, the tetrad forms. When the tetrad forms, something else happens often, and this is known as crossing over. So these homologous chromosomes, they have the same genes on them. Now, not necessarily the same version, because I said they're not identical, but they carry the same genes for specific traits. So let's say that we're talking about chromosome 5, and I'm making this up. I don't know what's exactly is on chromosome 5, but let's say that on chromosome 5, you would have hair color, eye color, and height. Okay. Chromosome 5 from mom will have hair color, eye color, and height. Chromosome 5 from dad would have eye color, hair color, and height. Same genes. Now, it doesn't have to code the same color. Okay. Dad could give you brown and mom can give you blue, or whatever. Okay. But the type of the gene is the same. Okay. So does that make sense? So that's why we say they're corresponding, they're homologous, but not identical, because the version of the gene could be different between the two parents. So each homologous pair is going to have the same genes, kind of the same areas. And so when they get close to each other like this, sometimes they overlap. And sometimes when they overlap, they exchange pieces between each other. And they exchange same regions between each other. On this image right here, you can just see like the tips being exchanged. It does not necessarily have to be just the end of the chromosome. You could have the middle section switch. And what this does is basically it ensures that genes from your mom and from your dad are getting shuffled. And it increases genetic variation. This is why siblings, unless they're identical twins, this is why siblings are not exactly the same, because they all are going to get a different set of your mom and dad's genes. Okay. So when you're forming your gametes, it is your mom's and your dad's genes that are getting mixed up and forming this new set that you're going to pass on to the offspring. Okay. So this crossing over only happens in prophase 1. It does not happen in any other area, and it mixes up, shuffles some genes of your parents in that cell. So that's prophase 1. After prophase 1, we would see metaphase 1 happening. Metaphase 1 meta means middle. Okay, so I want you to remember meta means middle. So these chromosomes will line up in the middle of the cell, and they will form kind of like 2 rows. Now, this is different from mitosis, because in mitosis, remember, those homologous pairs didn't really care about each other. So all the chromosomes in mitosis just formed one plane. They were all in one line. But here, metaphase 1 of meiosis, you have 2 lines of these homologous chromosomes. So that when the anaphase happens, what's happening is you're separating those replicated chromosomes there. Okay, so you're not separating sister chromatids yet. You're separating the homologous pair that's been shuffled, right, in prophase 1. So anaphase 1, you're separating the homologous replicated chromosomes, but not sister chromatids, all right? So that's anaphase 1. And then telophase 1 and cytokinesis is resulting with the cell membrane getting into pinching off, and nuclear envelope will reappear very briefly because this is going to go into meiosis 2 pretty quickly. So so now you end up with 2 cells, and these 2 cells now are already considered haploid because you do not have 23 homologous pairs. You have 23 replicated chromosomes, okay? But they're not homologous pairs anymore. And so we do not consider these cells right here. Okay, so this one and this one, those 2 cells are haploid cells already, and they are not identical. If you look at the colors of the chromosomes, you will see that they're not identical, okay? So that's meiosis 1. After meiosis 1, the cell is going to go into meiosis 2. Now, remember how I told you that in like before every cell division, the cell is going to replicate its DNA? Well, that's true, except for the steps between meiosis 1 and meiosis 2. So those 2 cells that you're seeing here at the end of this picture, each of them will go directly into meiosis 2 without replicating its DNA. It's not, after meiosis 1, there's no DNA replication, okay? It's just going to go directly into meiosis 2. And so let's look at our meiosis 2. And so here we're following those cells that we got on that previous picture at the end, okay? The order of the phases is the same. So you have prophase, metaphase, anaphase, and telophase, and cytokinesis. But you have to say that it's prophase 2, metaphase 2, anaphase 2, and so on. Because the events look similar, but they are quite kind of different. So prophase 2, your nuclear envelope that just reformed is going to disappear. The chromosomes are going to appear. Nobody is looking for anybody right now. You don't have homologous pairs. And then the cell goes into metaphase 2. Metaphase 2, you know, by seeing the chromosomes in a single line now. So in metaphase 2, you have a single line of 23 chromosomes, okay? In metaphase 1, we had 2 lines of 23 homologous pairs, okay? Or like 2 planes. Here they're in a single plane. In anaphase 2, the spindle fiber from the centrioles is going to grab onto the centromere. There's a region on that centromere that's known as kinetochore. I don't know if that's mentioned in your book, but that's what it's called. And the spindle fiber attaches to that chromosome from both sides, and then it pulls apart those sister chromatids. This is kind of looking like what you would see in mitosis. Okay, except in mitosis, you have 46 of these chromosomes in a single plane getting split apart. In meiosis 2, you only have 23 because we split them in meiosis 1, right? So anaphase 2, you separate the sister chromatids to opposite sides of the cell. And then at the end, you will have the telophase 2 and cytokinesis. Chromosomes are going to be enclosed in that nuclear envelope. Eventually the cell membrane will separate, and you will end up with 4 non-identical haploid cells. In the males, this is basically how spermatogenesis happens, okay? So for males, from that one cell, they can form 4 functional sperm. In females, this is kind of different because only 1 cell will result out of this process. The other 3 will become polar bodies. They will be very small. They will have barely any cytoplasm, and they will die. Okay, only 1 of the cells in females will keep majority of the cytoplasm, and that will be the one that can go and get fertilized. Polar bodies do not get fertilized. Okay, they die. So that's meiosis. Okay. I've mentioned that I would go over this because this I don't think meiosis is very difficult, but I just do want you to think about the differences between meiosis and mitosis. What is important is to remember that you do start with a diploid cell, and then how the tetrads form and all that. Be aware that before cell division, before the whole process, the cell is going to replicate its DNA. So you have 23 replicated homologous pairs of each, right? And so after the division, you end up with each single chromosome in a different cell. What is interesting to read is this clinical view 28.1. This is on non-disjunction. And this is where one of the chromosome fails to separate in the second part most of the time. So in that anaphase. So one cell, so like imagine that one chromosome is not separated, then one cell would have 2 copies of that chromosome. The other cell would have none. Like when you are looking at the telophase 2, if one chromosome didn't split in anaphase 2, one chromosome would have the whole replicated one, and the other one would be missing that copy. So if you have the cell that keeps the extra copy, and if that cell gets fertilized, then that's known as trisomy, because the individual would then have 3 copies of that chromosome. And the cell that is missing the copy, if that one is participating in fertilization, then that would be a monosomy, because it would not get a copy from that parent who gave that cell, right? They will only have copy from the other parent. So trisomy or monosomy. And you can see that, like, if you look at that image right there, you can kind of see how that would go. Okay. There are some trisomies and monosomies that are survivable, okay, but they do come with complications during life. So probably the most known one is the trisomy 21, and this is the Down syndrome, and the severity of the third chromosome there, and that's trisomy 21. So this individual has 3 chromosomes, number 21. There are different, like, it's a range of symptoms, and we don't really know like why some are milder than others, but that's known as Down syndrome. There are some other common ones that might go into pregnancy, and then eventually that pregnancy does not go all the way to the end, and those are trisomy 17 and 15 and 14 also is sometimes tested for. Is there 18 also? I don't know. But anyway, so but most of genetic mistakes like this are not compatible with life, so they're not survivable. And so that's that. Now with the X and Y chromosomes, I've mentioned that, like, normally the females are X and X and males are X and Y. It happens that if you have an extra X or an extra Y, that does not necessarily mean that it's not survivable, and the effects are also not as severe as with the extra copy of an autosome. So for example, we have females that might have an extra X or extra 2X. So there could be females that are triple X or quadruple X, rare cases even 5Xs. And these females are known as like super females, and that's not to say that they have special powers or anything like that, but a woman would not usually even know she has an extra X chromosome, because it does not interfere with any like mental capacity or anything like that. Females that have an extra X generally tend to look older, and they're a little taller. But again, you would not really know by looking or talking to someone that they are triple or multiple X's. Males could be X, Y, Y, Okay, so males could have an extra Y chromosome. This one has been associated with increased aggression in males, actually. Um, so having an extra Y is that, but again, there's no like other symptoms of that. Um, so there's a combination of XXY. That's a syndrome that actually does come with a um, mental delay. So that one, and also some physical characteristics. There are girls that are only X, they only have one X, and this is known as Turner syndrome. And there's no like mental delay or anything like that, but there are some physical characteristics that Turner syndrome girls have. They are generally shorter. So see, the women that have more X's are taller, and the Turner syndrome girls generally are shorter. They have a very typical shape of their eyes. It's not the same as Down syndrome, but the shape of their eye is affected, and some other physical characteristics. But I mean, mentally, they're completely fine, and just capable of everything that double X women are. So that's that. Uh, Although the Turner syndrome girls tend to be infertile because only having one X chromosome. So that's that. So that's our meiosis. The next section will be on female reproductive system. I will post the recording for that. I'm gonna go ahead and let you go, but I have a recording for the female reproductive system. This one is going to be a bit tricky when we're talking about the oocytes. Okay, so pay special attention to the oocytes and how that works. The good table for kind of knowing how this happens, or what happens when, is figure 28.6. So write it down somewhere, 28.6. There's also table 28.3 right before that, that kind of puts all the terms together for you. But I like 28.6 because it shows you the development of the oocyte and the development of the follicle side by side. So oocyte is going to be the cell that is going to get fertilized. It is housed in a follicle. So there are going to be several cells that are going to surround this oocyte, and so oocyte and the cells around it, that's follicle. Okay, so make sure that you know the difference between those two terms. And so that's what 28.6 shows you. And then I also like figure 28.7. 28.7 shows you the development of the follicle along with the hormones. So the top part, you see the follicle stimulating hormone and luteinizing hormone and what those two do. And there's that spike in the LH during right before ovulation. And then you can also see what estrogen and progesterone are doing during that time. And then at the bottom, you see the uterine cycle, what the uterus is doing as these hormones are changing like this. And you can see that the uterus, if you look at that figure 28.7, I don't know if you have your textbook next to you, but when you look at it, you will be able to see that the progesterone curve and the uterine growth curve are very closely associated. And so that's kind of a nice visual there for you to see what happens. And one other thing, I know I say that in the recording, but I can never say it enough times. The menstrual cycle is going to be like we talk about this in number or in average numbers. So we say that the cycle, ovarian cycle and uterine cycle, are lasting on average of 28 days. So there are actually not many women that have the cycle exactly 28 days. But if you average all the different lengths, it comes out to 28 days. So that's the number we're using. Be aware that this is very different from woman to woman. Okay, so the cycle is not the same for everybody. This is just kind of how in general it should work. The first day of the new cycle is the start of the menstruation. So start of bleeding is day one. It is not at the end. it is the beginning of the new cycle. So I will be asking you some questions on the exam that will be like, okay, does ovulation happen, or no, does no, menstrual flow happen before or after ovulation? And sometimes people want to say, well, it's at the end, so it's after ovulation. Well, no, within the cycle, that's the beginning of the new cycle, right? And if ovulation is day 14, then the bleeding or menstrual flow would be before ovulation, right? So remember, start of the menstrual flow is day 1, and then ovulation is about middle of the cycle, so 14, and then um, the other stuff, the um, development of corpus luteum and all that is going to be after ovulation. Okay, so pay attention to that part for the female. For the hormones, you just kind of need to know the general pathway. As I said in the beginning, you have the gonadotropin releasing hormone that stimulates luteinizing hormone, and then that stimulates the development of follicle. There are a few more hormones for females that you need to pay attention to, and you can see that in figure 28.8. So estrogen is important, and also there's going to be inhibin. Is it here? Yeah, and progesterone. And that is because in females the oogenesis works a little differently, again, from the regular process of meiosis. So females are designed only to, fertilize the egg once, and then if that egg implants, then they would carry pregnancy. You don't want to like fertilize any additional oocytes if there is pregnancy established. So it's very important that you stimulate some oocytes in the beginning. Generally, only one goes through the whole process of maturation, and that one is the one that's available for fertilization. If that one implants, then you're not stimulating another round of oocytes. Okay, so that's kind of the important reminders. But again, that will be on that in that female part. So I'll upload that female on to my TCC right now, and you already have male loaded there. On Wednesday, we're going to talk about development and pregnancy, and then heredity. I have to check that, but heredity, you'll basically go over heredity as you're doing that lab exercise. That's a handout. Well, handout upload. Okay, that's on my TCC. So, okay, so that's all I have for you today.